Okay, this is the Professor, CBMD PhD, and today's Monday, June 12th, 2023. It is 7:06 and 27 seconds, AM Eastern Daylight Time, and today's research, Actin filaments. So let's jump to it here. New study unravels the mysteries of actin filament polarity. Date June 8th, 2023. Source, University of Pennsylvania. An electron microscopy study revealed key details of actin filaments, which are essential structural elements of cells and muscles. Actin is the most abundant protein inside the cells of higher organisms, such as animals. It serves as a building block for long thin structures, called filaments, which provide key structural support as part of the cell cytoskeleton, the system that gives cells their shape and polarity. Rapid changes in actin filaments underlie key cellular events, such as movement along surfaces, cell-to-cell contact, and cell division. Actin filaments also are major elements in muscle fibers. The results of the study provide a mechanistic understanding of a process we have known about for more than 40 years, referred to as filament treadmilling, and impacts how we view the cellular roles of actin in health and disease. The dynamics of actin filaments are governed largely by the treadmilling process, through which individual actin proteins are shed from one filament end, known as a pointed end, and added at the other, barbed end. Actin filaments can be stabilized by distinct so-called capping proteins that bind to the filament ends to stop further addition or loss of individual actin proteins. Many other proteins also bind to the barbed and pointed ends of the actin filament. The structural details determining the specificity of these interactions, the details that explain why these two ends function so differently, have been murky. Researchers analyzed actin filaments using cryo-EM. With this high-resolution imaging technique, a researcher obtains many thousands of snapshots of a target molecule, aligns them computationally, and then averages them to reduce random image noise, yielding a 3D reconstruction of the molecule that may be sharp enough to visualize individual atoms. With artificial intelligence AI assistance, the researchers were able to focus on the ends of the filaments instead of their middle, as well as previously been the norm in similar research. By doing so, they identified hundreds of thousands of filament end views, allowing them to obtain near atomic scale reconstructions. As these revealed a flat actin shape or conformation at the uncapped barbed end versus a twisted conformation at the uncapped pointed end. The data also detailed the structural changes induced by the two actin filament capping proteins, CAPZ at the barbed end and tropiumodulin at the pointed end. As these are the two proteins found at the ends of the filament in skeletal and cardiac muscles, playing an essential role in stabilization of actin filaments in muscle fibers, and without these proteins, our muscles would fall apart. Results from this study provide crucial mechanistic details for a deeper understanding of actin biology as a whole. The researchers believe these study insights should also be helpful in understanding and ultimately treating disorders caused by actin dysfunction. And this is a professor of microbiology, bioinformatics, pathophysiology, and leader of research. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.